I want to hear a presentation. Yeah. So now I'm recording. Let's share the screen. Emre Hocam, your group joins not as much as before, right? I mean, there are not many people anymore.
Actually, 2 people, they have lecture at this time, so that's why they cannot join.
Ah.
And 2 people are preparing for the preliminary exams, so they are extremely busy. I don't know about the others.
Yeah, if you like, we can always change the time, as you know.
Well, since we are already halfway of the semester, I think no need to change it.
Okay, maybe one thing. So what I do is I put there's a section notes, and I put notes there. And when I make this presenter view, I have 2 screens, and in one screen
we can see it as well.
Yeah. Right now you see. I just want to show you. And here the next slide, so I can adjust my talk accordingly. So nothing is surprising anymore, and I have the notes with me. But yeah, now you are not gonna see it. Oh, I'm sorry. Okay. Yes. So this is today's article. The reason I selected. So what do you see right now? Okay, good. So I selected this article because I'm very much interested in seeds, and I wrote an, in fact, review about iron and seed. But in fact, I feel I feel myself not really confident about talking seeds. So that's why I chose a topic that was the most scary for me. I find this germination of seeds and ABA regulation quite complicated. However, we need to, now our research led us on seeds. So we need to understand, for example, yeah, we found iron deficiency prolongs seed germination, and we have no idea why. And we don't know how we can decipher it. So that's why we need to learn more about it. And for this reason, Babar was giving a talk about seeds and germination, and today I'm doing it. So seeds are important because they are the ones which give rise to the plant. And so seed germination is very important in agricultural sense. Seeds are expensive. If they don't germinate, then a lot of money is lost, and you don't have this plants, and you need to sow the seeds again and again. So seed germination is an important process. And in fact, it may look quite simple, but it's not. So first, let me tell you what seed germination is. So here is the seed. We have this embryo inside, and we have the seed coat. And these seeds are normally in a stage called dormancy. So dormant seeds cannot germinate. First, their dormancy should be stopped, and then they can germinate. Most of the seeds today, when we buy from stores, they are already non-dormant. But in nature, seeds do not germinate when they see water, because yeah, it would not be a good strategy. So they are in a dormant stage for a long time. They are like sleeping. They don't care if water is there or not. They won't germinate. Then germination is the process. So germination can be divided into 3. first, the water uptake. If you put the seed in water, then water will enter to the seed, and then it will cause the embryo to swell. So this is the second step, and then the root of the embryonic root, will begin to emerge. And when this root disrupts the endosperm and the seed coat, then the germination takes place. So taking water, swelling, and the rupture of the seed coat. So I talked about this dormancy thing. Let me make it more clear. So if you walk around after a rain, what you will see is a lot of mushrooms. That's because mushrooms absorb water and they just pop up. But seeds are not like this. So after a rain, when you walk around, you don't see a lot of seeds that are germinating. How do they germinate? How do they decide when it is appropriate to germinate? It's a very complicated process. So here you can see a lot of hormones and external cues are affecting seed germination. Basically, seed germination is dependent by these 3 hormones, although the others have effect on them. So the most important of those is ABA. Sorry, can I hear you right now, or do I have a problem with my... Can someone talk a little bit?
No, everything
Yes.
is fine. I think
Okay,
you can
good.
hear us.
Yeah. So ABA is the most important
Hocam,
of all. Yes. Yes.
Hocam, and there is a term of quiescent. Is there any difference between quiescent seeds or dormant seeds?
So the word quiescent, I only know this word for quiescent center, but I don't know. Oh, you mean
Hocam, there is a
dehiscence?
term for
You
seed
mean
quiescence.
no, dehiscence, right? Dehiscent. Quiescent or dehiscent?
Hocam, I don't know about it. I read it as a quiescence.
I think it's not correct. Maybe I can write it in chat. So I know this word, dehiscence. But quiescent is something else. I think you mix up, but I'm not sure. So dehiscence means, I think, maybe 2 things, either drying of the seeds or shattering of the seeds, quiescent. Emre Hocam, do you know what it means?
Now, I think the difference between the quiescent and dormancy is this. In dormancy, the seed actually in a state where even if you give water, it actually cannot start the germination process. In quiescent, I think when you give the water, temperature, everything required for the breakage or start of the germination, it can germinate. So you can count this quiescent as a state in which it is not really dormant, but it is still not in the state of the germinancy. It is in between. It is like a passage stage between the two, as far as I know.
Okay, so you think this area is quiescent area, right? So it's not germinated, but
yes.
It's also non-dormant here.
Yes.
Okay. Yeah, ABA is the most important hormone that decides the germination. We also have auxin and gibberellic acid. So the seeds, when they are shattered from the mother plant, have a lot of ABA. ABA puts the seed in dormancy, so it makes the seeds sleep. And gibberellic acid has the opposite effect. And auxin can have an effect through this path. And you can see under those hormones, we have many genes. Most of them are transcription factors. You can see some of them are called ABI1, 2, 3, 4. The reason for this is these genes are called abscisic acid insensitive. The way they found these genes was they grew seeds, they germinated seeds in agar that contains ABA. And when you put some ABA, then seeds will sleep, so they germinate much later. But some mutant seeds, they don't care if there's ABA there. So these are those seeds, those mutants. They get their name from the screening they are found. And they are, I think, mostly transcription factors. So here, among those, ABI5 is, I think, most important for us for today's paper. So ABA5 can stop germination, even if it is non-dormant, and even if it is almost germinating. So it's stopping the germination in the very late phase, as you can see. You can compare it with ABI3, which can also stop germination, but only if the germination stage is quite early. Okay, so this ABI5 is also under a lot of control, like the FIT. We already know for FIT there's so much control, but for ABI5 it's also phosphorylated, and it goes for proteasomal degradation. It's also under processes of nitrosylation or smolation and so on. Yeah, so this is how the seed germinates and all the network there. So there are a lot of more genes that have not been identified that are interacting with these genes. And today's paper will show us one of them. And it's called AGL21. What is AGL21? So the first real characterization of the gene was back at 2014, also published in Molecular Plant by the same group. So what they did was they showed AGL21 is involved in lateral root formation. So here you can see the overexpressor lines have a lot of lateral roots. Lateral roots means these roots that goes to the sides. And the mutants, when they lack the gene, they have less lateral root compared to the wild type. And secondly, what they found was this gene's expression is affected by a lot of environmental cues like jasmonic acid, ABA, and the other hormones. So their conclusion in this paper was, you know, root should have a architecture that is that should depend on the changes in the environment to make the best strategy to get the most of the nutrients. And this plasticity, so the ability to change root structure, is determined by AGL21. Okay. Yeah, so how did they come from this point to the germination? They just look where else is the AGL21 is expressed. And they saw, surprisingly, it's expressed in the seeds. And they know that in the seeds, there are no roots. So there must be a new function for the same gene. And they also know this gene is affected, the expression of the gene is affected by ABA, which is very important for seeds germination. It's the main hormone. So they smelt a new story here. So what they did was so easy and also clever. They just germinated the already available seeds, the overexpressors, the knockout mutants, and the wild type. And they just germinate very close to each other. But when they applied ABA, then they saw the mutants, the knockout mutants could germinate faster, whereas the overexpressor mutants could germinate slower. So here you can see they germinate slower, and here these two germinated faster than this one. So under pressure of ABA stress, the AGL21 makes a difference. Whether you have it and how much you have it makes a difference. Once you have the phenotype, now just go ahead and characterize it. So ABA was just one of a stress. In nature, we don't have this ABA stress. We have more real stresses, like salt stress or some other stresses. So what they did was next they examined if these seeds also give a phenotype under osmotic stress. Under osmotic stress, normally plants do not want to germinate because they think, oh, it's not a good time to germinate right now. So they just wait, this stress is gone. And when they did this experiment with the same lines, they also saw that under sodium chloride stress, so salt stress, and mannitol stress, which is also a type of osmotic stress, they see the same phenotype, which is the knockout lines germinated faster. So they made a mistake maybe because now they didn't think the environment is not suitable for germination. They just continue on germination. Okay, any questions about it? So this is just the
You
yes.
said that the plants wait for the right time to germinate. How do the plants know when is exactly the right time to germinate?
Yeah, they look for the cues, the external cues. They look for the water amount. They look for the temperature. And if, for example, temperature is too high, then this changes one of the genes expression, and then this then increases for example, ABI5. And then this water amount also, for example, is perfect amount, and then it decreases ABI5, and then everything is, you know, integrated in ABI5, for example. And the level of ABI5 in the end decides to germinate or not to germinate.
Is this in any way related to the plant's sense of time, like its circadian clock? Mind inserting. Like, is this related to the plant's sense of time and its season and what time?
So your question is if it is related to circadian rhythm.
Yeah.
I don't know. I guess everything is somehow related to the circadian rhythm, but maybe the question can be if circadian rhythm has a big impact on it. And I don't know. I think I haven't read anything about it. I think for germination, it shouldn't have very big impact, or it's not known, or maybe I'm not aware.
Hocam, may I ask something too? The osmotic pressure created by mannitol and sodium chloride differs significantly from each other, or they can be used interchangeably?
Well, I'm not an expert on it. So what I would suggest is, so if you have sodium chloride, plants will die. And if you look for the reason for that, I think if there is a lot of salt outside, then normally plant gets the water due to the solutes inside the cell. So the solute concentration inside is higher than outside. That's how the water passes and then goes up, right? If you have more salt outside, then you can get less water. If you get the idea, right?
Okay.
For mannitol, it's also, I think, similar. If you have mannitol, it increases the water it decreases, it increases the water potential like sodium chloride does, I think. So it holds water, I think, mannitol. So water is less flowing. Yeah, I'm not sure. I think, yeah, there will be less water flow to the plant due to this osmotic decrease in osmotic pressure with mannitol.
Now, Irem,
Yeah.
I think you have a very good question. And in order to understand this, please go to Can Kanzu's. I put his scholar link. go to his
Sorry,
link.
hocam. Thank you.
He has a publication, I think it should be in 2006, most probably. I don't
Bakıyorum
exactly
hocam.
remember the name of the paper, but it was actually giving a protocol for the application of sodium chloride, mannitol, and PEG-related drought stress, how you need to prepare the Petri dishes, what is the relation of the osmotic stress between those and how to do the calculations, etc. Look at that paper.
Hocam,
Really
there's a
good
paper named Methods and Concepts in Quantifying Resistance to Drought, Salt and Freezing, and Abiotic Stresses.
I think, yeah, that is that paper, yes.
Okay, thanks hocam. Thank you so much.
Seçkin.
Yes.
Now, by the way, if you are really planning to work on the germination effect of different stresses on the germination, I urge you to read the same paper
Yes.
for the design of the experiments. Plus, you should follow Can Kanzu's recent papers because he is the guy who found the SOS pathway mutants. Then he identified several very interesting mutants under abiotic stress conditions. And actually these ABI-related mutants are also coming from his publications. So it's quite important. Another thing in these type of experiments, especially if you are planning to deal with the root length or germination under these type of osmotic stresses, you need to understand the type of the media you should use. So I look at this paper, they are using the full strength MS media supplemented with, was it 1%, I think, sucrose. I think it was 1%. Now, if you read Can Kan's papers, you will see that this is too much stress already on the plant. Because of the sugar, it is affecting, giving some osmotic stress. As well as the minerals, amount of the minerals in the full strength MS is too much. That's why I think in this paper, they are already giving stress to the control plants as well. Majority of the people use quarter MS, and there is no sucrose supplementation for these type of experiments. So just read those. I think it will be really helpful in future.
Okay, thank you very much. So
Thanks,
nice to hear these discussions. Good. Okay. Yeah, so we have this protein ABGL21. It is affected by ABA-dependent pathway. So next thing was what the researchers want to know was if it is like very specific to ABA, or if its expression is also changed by the other stresses also as well. So they wanted to investigate how specific the response is.
Before we proceed, I have a question now. If they worked on, if they did this experiment only by using the knockout mutants, most likely they wouldn't be able to see a difference from the Colombia. And most likely they wouldn't do any other experiments. Just look, and interestingly, they only see the phenotype, germination phenotype in the overexpression lines.
Yeah, I'm not sure. I mean, yes, since they're not very expert on seed germination, maybe they can miss. But if you are an expert on seed germination, probably you wouldn't miss, because I mean, on the pictures it's not so obvious, but here I think it looks pretty obvious here, isn't it?
Well, it is not too much difference if you ask me.
Okay.
It is much more sharper when you look at the overexpressions.
I see. Yeah. I think you're right. So how we do our experiments with Babar is we also have a very small difference, but to make sure it really existed, we took so many precautions. So what we did was we amplified seeds several different times. So we use like 3 different batch of seeds. And it wasn't enough. We also used another organism like tomato seeds and Arabidopsis seed. They both show the same phenotypes. So only after that, yeah, we were convinced.
Okay.
Yeah, but I understand.
Sometimes if you don't see a difference by using one concentration of the stress condition, you may just try different concentrations, and I am sure at some point you will see a really nice difference.
Exactly.
It always
very
works.
important point. Yeah. Yeah. Okay. Yeah. Let's see. Sorry. Yes. So here, what they did was they examined the expression of ABGL21 in different stresses. And it was so interesting with ABA, jasmonic acid, auxin, and with other stresses, this gene expression increased. So it was responding to many different environmental cues. So it was not very specific to ABA, I think. But it's understandable also in the roots. It was answering all these things. So all these things somehow can change the expression of the gene. One interesting thing here is nitrogen deficiency. Under nitrogen deficiency, it also gives upregulation. And it might be interesting for us, Babar, because under iron deficiency, maybe, I mean, this gene also is changed, and this would be a new finding. We should analyze the expression of these genes in 1, 2, 3 days under iron deficiency. Yeah. So then, what they did next is important to understand. So, we have a gene in ABA pathway. So the next question is, where is this gene located at? Is it located somewhere in the middle or in the more upstream just after ABA, or close to ABI5, which determines this germination? But after ABI5, there are other genes like EM1, EM6, which we have seen also from Babar's paper. But where is our gene? From which place it makes an effect? To understand it, what they did was they took all these ABI mutants and they applied a stress like ABA, and they examined in which one of the mutants they see a different response. So this would show if there is a different response, I mean, if this response to the stress is changed in one of these ABIs, this would mean ABI is acting upstream of the gene. Because just think about this, stress is affecting if ABI is upstream of it, and if it is not there, then this thing do not get the stress. But if it was downstream, then this stress would affect this gene's expression. But so there wouldn't be any difference. Okay, let's come to the figure and then we can talk about it again. So they put ABA here in different mutants, 1, 2, 3 days, and then they saw all of them have the same pattern that they see for the wild type, except number 2 and 3. So ABI1 and ABI2. Okay, all the others, it was quite similar. So this means ABI1 and 2 are upstream of ABGL21, not downstream. And it was same for another stress they used, mannitol stress. They saw this regulation is always the same like wild type, except in ABI1 and 2, which shows they are upstream. Any questions about it?
I have one.
Yes, Anna.
It may not be very related to the subject, but I was wondering why there were significant changes in day 1, day 2, and day 3, for example, in mannitol or in IAA.
Yeah, I don't know. I would say, can you turn off your microphone? I would say this thing, ABI concentration and expression of genes during germination is very responsive. It's very responsive. It's a very dynamic process. I would guess it reflects the importance of decision to germinate, because you're a seed, you want to germinate, but you want to make sure the environment is perfect, and the environment, you collect all these information, and you need to really respond very effectively. So this requires, I think, a lot of expression differences. But other than this, I cannot answer you in more detail.
Hocam, I think these differences show the speed of the response of the ABI or the mannitol exposure, not exposure, but the plant's response speed to these stresses.
But the question is why under ABA, AGL21 is first upregulated, but then this upregulation doesn't continue but decrease. But in sodium chloride, for example, there is a continuous increase, but still, I mean, the third day is much more compared to second day, for example. Can you explain that? I mean, this is my understanding of the question. Yeah, by the way, why did they choose 2 stresses but not more? I guess having more stresses would be waste of money and time. And they chose these stresses, I think, because if you look at how the gene responded, we have 2 different ways. So these, these 2 respond similarly, and these 4 respond similarly. There's continuous upregulation, and here there's a little bit upregulation and a little bit downregulation. So they chose one of them from each, but doesn't matter. Okay, so next, sorry, I just gave these notes. Yes. Next, what we did was looking in more detail where ABGL21 is localized. I wanna tell you that it's quite advantageous to know this pathway of ABA very well. So we know a lot of genes. So that's why by just looking and comparing the expressions and using mutants, by just qPCR, you can locate where the gene is in this pathway. So we'll see how they did it. Yes. So here, what happens is they use the wild type, the overexpressor, and the mutant seeds, and they just examine the expression of ABIs and the other genes known that are in the pathway. So the logic is, if you have more protein, and when you have the more ABGL21, if it has an effect on the expression of other genes, it means all those genes are downstream of this ABGL21. But if increase or decrease of ABGL21 is not affecting expression of any gene, this means they are probably in the upstream. Try to understand this logic, really. I think it's very important. So here you can see ABI5 and all these genes. All these genes are regulated by the amount of ABGL21, in contrast with ABI1, 2, and all these genes. These genes, 4 of these genes. Okay, so these are probably on the upstream, and we know some of them are really upstream. And these are not upstream, but probably downstream. But where is it exactly? Okay, so in this slide, they did some experiments to pinpoint where exactly is AGL21. And how they did it was they took advantage of some in silico data and some indirect data. So let's look how the genes expressions are, some more of these, the other genes expression change in this pathway, so ABI genes, okay? And if you look at the expression changes of ABIs, we see some of them are not changed, definitely upstream, some of them are changed downstream, but one of them among all these genes changed exactly as AGL21 expression is changed. So it mimicked the change, which is ABI5. Just look at it. So when you have a lot of AGL21, you have a lot of ABI5. When you have little bit of or no ABI5, sorry, AGL21, you have less ABI5. That is why they hypothesized that maybe ABI5 and AGL21 are working together because their expression mimics each other, okay? So they wanted to further examine it. What they did here is they examined the expression of the seeds after imbibition to the germination. And what they saw here is ABI5 and AGL21 expressions are quite similar, especially when there is ABA inside the seed. Just look at it. So seeds before germination always have ABA, and we see after water is taken up, the expression of AGL21 decreases, which is same in the ABI5. And after germination, maybe it doesn't really reflect this, doesn't mimic, but when there's ABA, then we have increase of expression of AGL21 and also ABI5 too. So this shows when there's ABA inside the seed, so ABA pathway is activated, we see very similar expression of ABI5 and AGL21. So maybe they are working together, another data about it. And this is also in the same direction. So this reflects also ABI5 expression because it's under ABI5 and we see a similar expression pattern. And here in E and F, what they did was they just entered to a website where you can see the general microarray data. There you can find out expression of each gene during developmental stages. And they looked at the seeds. So here seeds are developing inside the sleek, you know, and here they are mature seeds. And even in here and also here, this is the expression of imbibed seeds, also in silico data. Just they entered the data on internet and they found out this. So here you can see also the trends are quite similar. Not only in their real experiments, also in silico data, they mimic each other. Okay, so they thought now it's time to do also protein assay to look if proteins are also behaving similarly. And when ABA is added, so pathway is activated, they saw that the overexpressor lines have more ABI5, wild type has less, and AGL21 has even less, which means the AGL21 expression determines how, not only how much ABI5 transcripts are there, but also the protein level. Any questions until now?
I have a question.
Okay.
Yes.
Now in this G, Western blot, why do you think they observe two bonds in ABA minus?
I would say, I think maybe the antibody is not only capturing ABI5. This is one possibility. Maybe there are some other, well, not the other ABIs, but I think maybe some other genes which are similar to that, or maybe there is alternative splicing. Yeah, this would be my answer.
Okay. You talked about the phosphorylation, ubiquitination, all those things.
Yeah, you're right. Some part of it maybe is phosphorylated, whereas part of it is not. Yeah.
I think so. I think
Very
that is
good idea.
that
Yeah.
because they used monoclonal antibody. So it is specific to ABI5 only. But that is interesting to see that this is really working nice.
Yeah, very good point. Okay, İrem?
Hocam ben de aynı şeyi soracaktım.
Vallahi. Okay. So from now on, I think they do more experiments, but it's mostly to fill the thing. So it seems to me obvious that ABI5 and AGL21 works together. So,
Hocam,
yes.
in the last graph, they also showed ABI4 and 3 kind of showing a similar pattern to ABI5 in the A part of the graph.
Sorry, are you talking about A?
Yeah, I'm talking about A.
Okay.
ABI4 and 3 also showed a similar pattern to ABI5.
ABI4 and 3. I don't think so.
I mean, there's an overexpression when there's an overexpression of ALG21. There's more ABI4.
Yes.
And when there is less AGL21, there is very much less.
Less compared to the overexpressor, but you should compare it to wild type.
It's, I guess, yeah, but it's not a significant. It's maybe like a 0.3. Maybe.
No. What I mean is this one should be lower than this one. This one should be lower than this one.
Oh, okay. But why do you think it's more overexpressed in the overexpressed version? Like, why is it?
No. This is wild type where you have AGL21.
I know, but I'm saying, why is it more in the overexpressed if it is not directly correlated, directly co-expressed?
Sorry, I didn't understand.
I'm saying, why is ABI4 and ABI3 also having a spike when the AGL21 is
Yeah. I understand now.
Even if it's not strictly co-expressed, correlated.
Yeah. I think there might be many reasons for that. We see there are a lot of loops, feedback loops in cells. So if one protein the number is increased, it may signal the other proteins somewhere else to degrade. I think it should be related with that. So it might have been still possible that these things also directly interact somehow. But if you ask which is the most likely, this would be the most likely one, I think. Okay, good. Good questions. Okay, so here they took advantage of the mutants, and here they didn't examine the molecular level, but instead the phenotype. So the overexpressors, as we have seen, grows slower, and ABI just a second. Yeah, the overexpressors grows slower, and the ABI5 itself grows faster. Okay, the mutant grows faster, and this is slower. So when they cross these two lines, if they act separately, what they should find is a plant which would grow not as fast as this one, but not as slow as this one. So in between, right? So let's see what they found. So they found this line grows the same as ABI5, which means AGL21's slowing effect, germination slowing effect, can only be possible in the presence of ABI5. Otherwise, it doesn't have any effect. So ABI5, it works maybe with ABI5, and without ABI5, it doesn't do anything, not up, not downstream, nothing. Okay, so its effect on germination is dependent on the presence of ABI5. This is an additional evidence that they work somehow together.
Now, before we proceed, sorry,
Yeah.
I think we need to ask a few people if they really understood your description.
Mm-hmm.
Because some of them, for instance, Irem, are looking really puzzled. They are trying to understand what you just said. So maybe
we should
we
slow
can
down.
ask one of them to paraphrase that.
Yes, please go on.
Yes, Irem, would you like to say a few words?
Hocam, now I understand that they co-express together, and they are, I mean, necessary for plants to grow better, but I don't understand the molecular logic exactly.
Irem, are you aware this one doesn't have ABI5?
Yes, I'm aware.
Okay, good. And this one has a lot of AGL21.
Mm-hmm.
Okay, maybe Cem, can you describe the figure?
Hocam, I am also trying to understand now.
Okay, I'm sorry.
Hocam, can I try?
Yes, Onur, please.
Actually, I understand that ABI5 and AGI21 work together. We supposed to see that thing. When we observe AG21 in overexpression, it's grow very slow.
Mm-hmm.
But there is when there is no ABI5, we observe a very fast growth. If they don't link together, we expect to see that in between the ABA1. Actually, I need to use the annotation.
Yes.
Okay. We need to see this one in between these, not that much slower than AG21 overexpression, not that much faster than the ABA5, but they work when we observe the phenotypes. It's much more similar the absence of ABA5. So we can understand that how whether there is a lot of AGL21 or not, AGL21 couldn't work well without ABA5.
Yes. To make it very short, I'll tell once more, and then we can ask one more time then. If we have AGL21 protein, then it slows down germination. But if ABI5 protein is not there, the transcription factor is not there, then it doesn't slow down the germination. Which means this slowing down effect, the effect of this protein, the presence of this protein is only meaningful if there is ABI5 protein. Bir analoji yapmamız lazım burada. We should make an analogy here so everyone understands better. I'm very bad at analogies.
Hocam, is it something like epistasis?
Yes, I think it's called epistasis, which means presence of one gene changes the expression of another one. Is it what epistasis means?
Yeah. If, for example, AGL21 does not exist, then ABI will not be able to do what it needs to do.
Yes, maybe. Okay.
Now, can we ask the same question to Amir? Amir, if you can listen us, can you explain this figure?
Actually, Jam, I didn't read the paper.
But when you look at this figure, and
Yes.
especially the graph in ABA treatment, can you tell if ABI5 or AGL21 is upstream? Which one controls the other one?
It's looking same, Hocam.
No.
Yeah,
You're asking about under APA treatment or under control, Hocam? Yeah.
No, just look at the ABA.
ABA, okay. Yes, AGL is down.
So you think that ABI5 is upstream of AGL21, right?
Yes. Under ABA, yes. Under ABA treatment, it's upstream. But in the control,
Okay,
there is,
now I would like to ask the same question to Babar. Babar, do you agree with Amir?
No, he just said the reverse.
Then can you explain the correct version?
Yes, actually, AGL21 is the gene that is controlling the expression of ABI5.
And can you, just by looking to this image and graph, can you explain why do you think so?
Yes, because when we overexpress gene, it is helping ABI5 to suppress the germination. But when we are knocking out this gene, it is reversing the effect of ABI5. So germination is improving. So it is not able to switch on ABI5, which blocks the germination.
Yeah, but we don't have all those information just from this graph. So can you tell us what the graph says? I mean, without any information like molecular, there is no molecular information here.
Yeah.
So here, what I can say is that when we overexpress this gene, it blocks the germination, and when we knock out ABI5, it improves the germination.
Yes, it makes the germination faster when you knock out ABI5. Yes. Okay. And then when they are together?
When they are together, overexpression is more prevalent as compared to knockout of ABI5.
What do you mean?
I mean is when it is being overexpressed, somehow it is maybe it is interacting with other genes.
No, you cannot say such a thing from this graph. You are actually just trying to say something extra. Now look, the main idea of this experiment is actually looking to the genetic interaction between the two genes. That's it. And according to this interaction, first you need to look at the phenotype of the single mutants. So in ABI5, we have high germination rate. In overexpression plant, we have low germination rate. And then when we make a double mutant plant, you can think the middle one as the double mutant. In this case, it looks similar to the ABI5 single mutant. This indicates that ABI5 is downstream of the AGL21. Why? Even if you overexpress the AGL21, if there is no ABI5, you don't see a phenotype. It is similar to the single ABI5 phenotype. This indicates that ABI5 is downstream in the pathway. Do you understand this logic?
Yes. Yes.
So actually, the interaction between the two loci is understood by doing this type of experiment. And this is a perfect experiment to show which gene is upstream and which gene is downstream. This experiment only tells the location of the gene as compared to the other one. That's it. Now, as you can see, we know that AGL21 is upstream of the ABI5. That's it.
Yes. Okay. I move on.
Please.
So if this AGL21 is upstream, then it would mean two things: whether it binds to ABI5 directly, or maybe there are some other genes in between, and then it binds to another gene, and this gene somehow increase expression of this one, ABI5. So to examine whether AGL21 is able to bind ABI5 or not, they did qPCR, chip qPCR analysis. So this is the promoter sequence of ABI5. And here they know AGL21 type protein belongs to a group of proteins like METS-Box protein family, I think. I'm not sure. And it's already known what type of sequences these METS-Box proteins bind. And when they examined the promoter region, they came up with 5 different regions where this gene is likely to bind. And then they did the chip enrichment analysis. So I'll repeat once more how it works. Please try to understand it. So what they do is they overexpress they have this line, AGL21 overexpressor line with H8 tag. So they overexpress it in the plant, and then they crush the plant, and they put a chemical which fixes all the proteins where they are. So if some of the proteins are on the DNA, bound to the DNA, they will stay bound. If they are outside, they will stay outside. Then what they do is they have antibodies, HA antibodies. They take only the proteins. So all the rest of the DNA, RNA, everything is outside. They only have all the proteins. So if now their protein is bound on the DNA, is able to bind on the DNA, then they have this DNA with them in their mixture. Now they do qPCR. Which DNA regions are more in their protein mixture? So they designed 5 different real-time primer sets. They observed these regions, and they found out that all the regions are represented equally except P5. So from this region, DNA is much more in their protein mixture, which means AGL21 can interact ABI5 from P5. So here we can see this is enriched. This region only is enriched.
May I ask a quick
Yes,
question
please.
from B? Now, in the P5 section, after the addition of the ABA treatment, there is a little bit more increase. Did they mention any difference, significant difference between the normal control and ABA treatment? I'm not sure if there is a really difference between the two.
You're asking if they discuss this result, right?
Right, if that two is different.
Yeah, because they didn't examine if they are different. There is no statistics about it. I now got you. I think they don't discuss it, and no, they didn't examine it.
Okay. Okay.
Yeah.
Ajam?
Yes.
About the chip qPCR thing, when the protein is left, when they take protein is left and there's some DNA in their protein, do they know specifically which protein bound to which area of the DNA, or are they just kind of like, we have this amount, these proteins and these DNA, like we have this like region of the DNA, but they don't know which one bound to which?
Okay. I'll tell again, maybe more clearly.
No, I understand that there's the DNA is bound to the protein, but when the DNA is left with the protein, do they know which protein is bound to which area of the DNA?
But here they only take one protein, not a protein mixture.
Yeah, I mean, but usually about the process, not in this specific instance. Do they know specifically which protein bound to which specific region, or are they just estimating on the level of protein in the DNA?
In chip PCR or just in silico data? Are you talking about A or B?
I'm talking about A, how they have
okay.
like regions.
Okay, now I got you. So I think how they know it is from the experimental data. They have this matchbox protein, they characterize it, and then how they do it is in one paper, they look for this interaction, and the way to do this is they change the amino acids and they examine every time again if these things bind or not. This is one way they do it. There are other experimental ways to identify these cis elements, and once these cis elements are identified, for example, they can examine them in different species and find out if they are conserved. If they are conserved, if some nucleotides are not conserved, they may say, these nucleotides are not so essential, so they can be changed. This is how they come to these sequences. Is it answering your question, or do I misunderstand it?
No, Jim, answers a good part of my question.
Okay, good. I'm happy about it. I have great difficulty to understand questions. I don't know why. Yeah. Also, when I give lectures or talks, in the audience, people ask questions, and I have really great difficulty understanding them. I don't know why. Emre Hocam, can you give me any suggestions about it?
Now, if you don't understand, you can ask them to paraphrase it.
Yes, with different words.
If you after the paraphrase, if you still do not understand, what I do is I give a general answer, and then I look at their faces. If they are not satisfied, then I say that after the talk we can talk in detail.
Okay. Yeah. Okay. And here in C and D, what they did was just now I'm a bit disconnected. Give me a second. Yes, they activated the ABA response, and yeah, they look at expression of EM6 and EM1, which is the we know it from previous papers like Babar's paper also. It shows how much ABI5 is expressed. So if ABI5 is expressed a lot, if we have ABI5 protein a lot, sorry, not expression, but protein, we tend to see higher EM6 and EM1. So what they did here is in the overexpressor of this gene, we expect to see more ABI5 and we see more EM6. In the mutant, we expect to see less and we see less. And in the double mutant, not double mutant, but when you express them together, yeah, I'm confused here. Can anyone help me?
Hocam, it is like the previous figure. If there is no ABA5, the overexpression cannot express that much higher.
Yeah, it cannot increase it, right? Yeah, you're right. So we should look at here. Sorry about the confusion. So these two are equal. So normally, this is just the repetition, I think, of the previous experiment in the previous slide. But now we are not looking to the phenotype, but we are looking in molecular level. Yeah, I think this is just to fill some more space. I think it doesn't give more information. But now the number of AGL21 protein is not important, provided that ABI5 is not there. They can only work together. Okay. So any questions, or I skip to the last slide, which is the... So this is the slide I showed you at the beginning in the introduction. So they were searching for a new gene, AGL21. They know it's involved in ABA response. What they didn't know was where to put it. And they found out that it directly interacts with ABI5. I mean, we should say it directly changes expression of ABI5, thus changes the germination process. And itself, AGL21 itself is in fact affected by a lot of hormones and so on. So we should also put some arrows here. I think I like this article quite a lot. The reason is I think it's very typical. If you know all the pathway, when you see a phenotype, just do real-time PCRs to find out in which step it can stay, it can sit, you know. So it's very simple approach. I like it for this reason. And second reason I liked it is from the same group that, I mean, they published in two very high impact journals about the same gene. They already have everything. What they did was just looking to the expression of the same gene in another place and then start a new story. I guess this story could be completed like in 1.5 years easily. Okay. Thank you for listening. Please ask me questions if you have.
Hocam, I have.
Babar, continue. I will ask later on.
Yes.
Hocam, I have just added another germination paper in Journal Club Plan, so you can check.
Okay, thank you.
Now, I have a question.
Mm-hmm.
So last week's paper was published in a journal of impact factor around 4 or 5. And in that paper, there was a lot of qPCR physiological tests, as well as there was the same chip experiment, EMSA experiment, everything.
But wasn't it phyto? What was the journal it was published?
It was integrative something.
Are you sure?
Journal of Integrative Plant Biology. I am just looking at it now.
Okay, okay.
It has the FETRA 5.
Amir's paper, right? No.
No, that was Abdullah's
paper.
My
Okay,
paper,
okay.
my
I
paper.
get it. Yes.
It was in the online.
And now, so in that one, as you can see, they were trying to integrate circadian clock into the iron deficiency, iron homeostasis. But now here, when you deal with the germination and you do actually less experiments compared to the other paper, and you publish in a journal impact factor of 12, 13, I don't know how much. This molecular plant is really high now. Why do you think this is the reason? Or what is the reason for this? Do you think people would appreciate working on germination as compared to the iron deficiency?
I would say so. But also, I think this I forgot the name.
Oscillators.
Yeah, I mean this whole thing, the complex. I think it's not in the very center of iron deficiency, and also iron deficiency is not at the very center of the oscillations and so on, this stuff. But here, I think this protein is really at the center. It changes ABI5's regulation, and ABI5, I think, is the most popular gene in about this germination thing.
And actually, although Abdullah didn't mention while he was presenting that the interaction between the circadian clock and iron homeostasis has already been studied. The first paper was published in 2000, I would say 13, most probably.
Yeah.
So this was like extra paper. That was the reason. By the way, working on ABI mutants is extremely difficult. It's horrible. I used to work on some of them and I couldn't work. That's why I stopped working with them. The reason is that because of the problem in the ABA concentration in those, unfortunately, in some mutants, I forgot which one, I think it was something like ABI1 and maybe ABI3, that some mutants can, you should not wait until the seed becomes a dry, dormant seed in order to start your experiment. For instance, if you collect the seeds, that those seeds, like the ones look like Colombia, then they will never germinate. Wow. They have these type of problems. And most probably in many of these experiments, they were not using the normal seeds we know, but instead they were using the seeds that have not been dried already. You just need to collect the seeds from the plant and immediately you need to start the experiments. So that's why it is extremely difficult. And doing a crossing, like most probably in this overexpression line cross to the ABI5, most probably they were doing a crossing, or I don't know if they did the transformation. If it is crossing, screening of the F2 is extremely difficult. It's almost impossible because of the mutant phenotype. So that may actually let them publish in this journal.
Okay. How do you know so much about germination?
I worked on it.
I didn't know that.
My PhD, now starting, although my PhD title is related to iron and cadmium, starting topic was salinity. The mutant I was working, we first characterized it under salinity, drought,
CPL
cold,
mutant,
CPL.
right?
And then we did microarray and we found genes related to iron deficiency. Then we switched to iron deficiency.
Ah, okay.
But my other papers related to this mutant, they are all about the cold, different other stresses. We did a lot of other things with some other. So that's why I was talking about Cankan. We worked together with his group.
Okay, good to know.
If you are planning to do some other experiments related to germination,
Yes.
or something related to the ABA signaling, gibberellic acid signaling, let me know.
Yes, yes. We should discuss with you with Babar also. Yeah, but we need more time to understand what's going on, and then we'll come to you.
Okay.
Yeah, thank you very much for your attention. Maybe we can leave now. If you don't have any questions. If you have questions, you're welcome. We still have a lot of time.
Hocam, can you repeat a little bit about the in silico experiment that they did?
Yeah, I can show you. I don't want to repeat it by just saying it, but I want to show it to you. So here, you see my desktop now, right? So here you write the name of a gene, 83G58660. This is MTP8. And then you push search. And here, if you go to EFP browser, maybe this one. Sorry, this one, EFP browser. Then they show you how much it's expressed in the developing seed. So the darker the color, the higher the expression. Here you can see these are the seeds in different stages, starting from 3 to 10. 10 is the mature seed. You can see my gene, this MTP8, is very much expressed in the late stage. And then what they did in the article, they put another gene also here, and then they compare these two genes. So we see here the color, but it's possible to reach to the numbers, and they put the numbers and they make the bars.
Hocam, is it coming from the data we discussed a little bit in March? We saw from an article from the Nature.
I don't know. I think not, because this data comes from plus one article from 2007. There are many of these, so it's unlikely we talked about the same.
Okay.
Any questions? Okay, next week is Amir's presentation, right? We're looking forward to hear it. So bye-bye. See you later.
Bye-bye.
See you.
Thank
Bye-bye.
you.
Take care.
See you.